Good afternoon. Today we are looking at meiosis. So, what is meiosis? Meiosis is a similar process to mitosis, however has a very different function, only occurs in specific cells. This occurs in the germline of both male and female bodies which undergo meiosis to form gametes by a process called gametogenesis. So, in spermatogenesis, sperm are created in a continuous and prolific manner. Both daughter cells from the germline produce functioning sperm. In oogenesis, egg cells are created during embryonic development by a prolonged process and do not reach the maturity for many years. The daughter cells are unequal, as one daughter cell receives most of the cytoplasm, causing the second to degenerate. Chromosomes. So, human somatic cells have 23 pairs of chromosomes. The two chromosomes in each pair are called homologous chromosomes or homologs. The chromosomes in a homologous pair are the same length and shape and carry the genes controlling the same inherited characters. The sex chromosomes which determine the sex of the individual are called X and Y. Female bodies have a homologous pair of X chromosomes, XX. Male bodies have one X and one Y chromosome. The remaining 22 pairs of chromosomes are called autosomes. Each pair of homologous chromosomes includes one chromosome from each parent. The 46 chromosomes in a human somatic cell are two sets of 23, one from the mother, one from the father. A diploid cell 2N has two sets of chromosomes. For humans, the diploid number is 46. 2N is equal to 46. A gamete contains a single set of chromosomes and is thus known as a haploid cell. For humans, the haploid number is 23. N is equal to 23. Each set of 23 consists of 22 autosomes and a single sex chromosome. In an unfertilized egg, ovum, the sex chromosome is X. In a sperm cell, the sex chromosome may be X or Y. Look at this diagram here. It will show you meiosis, fertilization, mitosis and development, and the haploid and diploid cells. So, meiosis occurs via just the same way as mitosis, the application of chromosomes. It has two consecutive cell divisions, meiosis 1, meiosis 2. The two cell divisions result in 4 daughter cells rather than 2 daughter cells in mitosis. Each daughter cell has only half as many chromosomes as the parent cell. So, this is how it occurs. Interphase, meiosis 1, meiosis 2. So, interphase, pair of homologous chromosomes in a diploid parent cell. Chromosomes duplicate. You get a pair of duplicated homologous chromosomes, sister chromatids, and a diploid cell with duplicated chromosomes. Meiosis 1, the homologous chromosomes separate, haploid cells with duplicated chromosomes are produced, and then the sister chromatids separate, and haploid cells with unduplicated chromosomes. So, let's go through this step by step. 4 phases. Prophase 1, Metaphase 1, Anaphase 1, Telophase and Cytokinesis. And this is meiosis 1, remember. So, prophase 1. This involves crossing over, which produces recombinant chromosomes, which combines DNA inherited from each parent. The crossing over contributes to genetic variation by combining DNA from 2 parents into a single chromosome. In humans, an average of 1 to 3 crossover events occurs per chromosome. So you see here in prophase 1, each chromosome pairs with its homolog and crossing over occurs. So homologous chromosomes are aligned, chromosome crossover, recombinant chromatids, and non-recombinant chromatids. Metaphase 1. In metaphase 1, pairs of homologs line up at the metaphase plate with one chromosome facing each pole. In anaphase 1, pairs of homologous chromosomes separate. One chromosome of each pair moves towards opposite poles, guided by the spindle apparatus. In the beginning of telophase 1, each half of the cell has a haploid set of chromosomes. Each chromosome still consists of 2 sister chromatids. Cytokinesis usually occurs simultaneously, forming 2 haploid daughter cells. To summarise meiosis 1, interphase, prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis. Meiosis 2. Meiosis 2 involves prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. Meiosis 2 is very similar to mitosis. So, in prophase 2, a spindle apparatus forms. Chromosomes, which are each still composed of 2 chromatids, move towards the metaphase plate. In metaphase 2, the sister chromatids are arranged at the metaphase plate. Due to the crossing over in meiosis 1, the 2 sister chromatids of each chromosome are no longer genetically identical. In anaphase 2, the sister chromatids separate. The sister chromatids of each chromosome now move as 2 newly individual chromosomes towards opposite poles. In telophase 2, nuclei form and the chromosomes begin decondensing. Cytokinesis separates the cytoplasm. One summary. Property: DNA replication. Mitosis, which occurs in both diploid and haploid cells, occurs during interphase, before mitosis begins. In contrast, meiosis only occurs in diploid cells, and it occurs during interphase before meiosis 1 and not before meiosis 2. Number of divisions: 1 for mitosis, prophase, prometaphase, metaphase, anaphase, telophase. 2 for meiosis, including prophase, metaphase, anaphase, telophase. Remember meiosis 1, meiosis 2. Yeah, so prophase 1, prophase 2, metaphase 1, metaphase 2, anaphase 1, anaphase 2, telophase 1, telophase 2. Number of daughter cells and genetic composition: 2 for mitosis, each genetically identical with parent cell with the same number of chromosomes. For meiosis, 4, each haploid genetically different from the parent cell and 1 from each other. That's the end of today's video, quite a short one today. Thank you very much for this. I look forward to seeing you all in the next video. Please do comment, like, share and subscribe, and thank you very much once again.